What we discover in the lab can then be moved on to patients to say, right, we need to stimulate this group of neurons to ameliorate anxiety or PTSD, for example. That would be extremely satisfying. I'm going to briefly introduce myself. So my name is Tom Mrsic-Flogel. I'm a neuroscientist and a biologist by training. I was born in Croatia, and then when the war started in Yugoslavia in 1990-91, I moved to the UK to do my undergraduate PhD, and then I went on to do further training as a postdoc in Munich, Germany. I started my lab at UCL in 2007 in somewhat of a decrepit building, unlike the one you saw earlier or here on the top right. And then I was offered a place to go to Switzerland, and then finally an opportunity arose to lead the Sainsbury Wellcome Centre in 2016, where I've been ever since. Okay, so briefly, I think Georgina already introduced this. The Sainsbury Wellcome Centre is a brainchild of the Gatsby Charitable Foundation and Wellcome. They both funded neuroscience for long periods of time a priori, and they decided actually it was maybe a smart idea to really, it is the right time in history to really give their push. Instead of funding individual labs, let's create something magical and perhaps something more synergistic by co-localizing various types of experts in the same building. And the mission was set at the start to simply explain how brain gives rise to various aspects of what we call behavior, but by that I mean cognition and perception, how we act and how we learn. The inaugural director was John O'Keefe, so he was appointed in 2014. And then currently, the Sainsbury Wellcome Centre has approximately 190 individuals working in it. That includes scientists, non-scientists, etc., of which there are 12 group leaders, so they're the ones that run laboratories. Now, co-localized in the same building is the Gatsby Computational Neuroscience Unit, or Gatsby Unit for short. And their job, unlike our job, is to provide mathematical descriptions of intelligence and different aspects of behavior, again, perception, cognition, action, and learning. And it's a fantastic idea by Wellcome and the Gatsby Charity to actually put us together, because it is at this intersection where we hope to do great things. Now, just to kind of give you a sense of that, so for example, the most recent Nobel Prize was awarded to Jeff Hinton, who was the founding director of the Gatsby Unit, and Demis Hassabis, who was a postdoc there. And for example, Demis Hassabis founded from the Gatsby Unit Google DeepMind, that now went on to transform, basically created the new field of AI and transformative things it's done, including things like AlphaFold, which has great benefits for medical treatments. So new building. And together, if you want, our joint mission to understand the principles of intelligent behavior, both in brains and machines, and the two fields will feed off each other, and we can discuss that perhaps at the end. Okay, so what is SWC's vision and strategy? Put it in less formal language, what we do and how we do it. So the mission is to understand how the brain gives rise to complex, flexible behavior. I'll unpack that in a bit. And this is an incredibly hard challenge. I will explain to you why is it a hard challenge. But that's why it's all the more important to bring together co-localized, world-leading experts that also want to collaborate, not just that they're smart, but also that they want to work together. We think also that our job is to train the next generation of future leaders, because solving the brain and intelligence in general is a hard problem that we may not solve in the next 10 or 20 years, but we want to kind of create the next generation that will do so. Many of the things that we do require the development of new tools, technologies, software, etc. So we actively develop those and share them with the rest of the world, because again, it's not just an endeavor that we do in the building, it is also happening at the same time in many places. But for our science to flourish, it's crucial to build an environment that is collaborative and supportive and diverse, where everyone can contribute their bit and everyone feels they belong and everyone has the same sense of mission, and we can discuss that a little bit in part of the research culture. And we have been from the outset very keen in engaging diverse communities, bringing in all kinds of voices and being very active in bringing people on board, teaching them about various aspects of AI, machine learning, neuroscience, and bringing those younger individuals on board. And we hope that collectively through these efforts we'll impact society through our discoveries, technologies, innovations, and so on. Okay, so let's go to the scientific questions, which is what my talk will be about. We'll pick up on the research culture later. So we want to understand a hard problem, how our brains enable us to perceive the world, reason, plan, control our drives and states of mind, make decisions, how they learn, all in the service to interact flexibly, and the key word is flexibly, with the world around us. So if you think of it from the evolutionary perspective, that all brains do is they flexibly control behavior. Whether it's humans or animals, that's all we do. The only output is how we move in the world, how we interact with other objects or other individuals, or indeed, and speaking is one just way of doing that. So how do we think about this problem? It's a pretty hard problem. So the way we think about it, and not just we as neuroscientists, but also the AI community, is that brains or machines build an internal model of the world. They're trying to map the relationships that are out there and store it somehow in the connections and in the networks, such that we can predict what will happen next. And that internal model is extremely powerful. We can do simulations, we can do best guesses what's out there. And I'm going to give you a few examples of that. So here's one example. I believe that you all perceive that this is a square. There is no square there. There's no outline of a square, but your brain fills in and guesses that this is a square. And that's because you've encountered many squares before, and if some bit of the square is occluded, you simply interpret it as a square. So it confabulates the existence of a square. And these internal models, this is a demonstration that these internal models of the world that you've learned through experience dominate our perception. Excuse me. Right, here's another example. This is more a fun one. This internal model of the world about how you perceive the world could be extremely rapidly updated, okay? So I'm going to show you an image, and if you don't tell me what it is, and I'm going to show you a real version of that image, and then let's see what happens. So this is the image. Now, most people who see this image for the first time cannot recognize what's on it, okay? Now we're going to give you a brief flash, and now probably most of you cannot unsee the frog. So in less than a second, your brain has irreversibly changed such that next time you see the same image, you will see the frog. And that's because now the input coming into your eyes from this image is exactly the same. It's just something in this instant change in your brain that helps you interpret it. And that is updating of that internal model that I talked about that dominates your perception. Now your perception of this is completely different than it was, a minute ago. This same internal model not just dominates our perception, but also helps us reason. Now this is a simple Lego house, but based on that, you can actually infer lots of things about what's going on here. If I tell you, is this a family house and does the family have teenage kids? You can say, yeah, because there's a basketball hoop out there. If I tell you what season is out there, you can say, well, it's probably spring or summer because there's a lawnmower parked on the lawn outside and you don't mow the lawn in winter. You can probably tell me that the bedrooms are upstairs or the kitchen is downstairs. All this stuff comes from your prior experiences where you extracted some knowledge about the world, and now you can infer things about the world that you haven't seen before. You've never seen this Lego house before. You can tell me lots of things about it if I ask you questions. Now, there's this kind of perceptual reasoning part of this internal model, but then we use that model to act. We make decisions, countless decisions every day, and we plan and reason. So this is just an illustration that we can not only act things out, but we can make a plan and then act things out. And so a lot of what we do is studying how that is done by the brain too. Including things like abstraction. If I tell you you should build this Lego house, you can do it in a compositional way. You can say, first I have the bricks, which are the minimal units. Then from those bricks, I will assemble doors and windows. And then I'm going to have a plan in my head how I'm going to assemble the house or whatever you choose it to look like. Okay, so this is a very flexible model. And if I tell you, well, make the house a bit longer, a bit taller, you'll be able to do that very easily. This flexibility is a hallmark of intelligence. Now, how do we build this model? I'm sorry for the technical nature of this slide. Is the key driver behind building this model of the world is learning. And now we've known, both from AI and real brains, that there are 3 different flavors of learning in the brain. One type of learning is called unsupervised learning, where your brain discovers structures in the world without being explicitly told what to look for. This is, for example, how babies learn language. They hear a stream of frequencies that are coming from the mother or the father, and then they chunk up those frequencies into words, syllables or words, such that the words become units, and eventually they start making sense, right? So we break up the structure, the statistics of the world into meaningful chunks. Now, the way you give additional meaning to these chunks is, for example, through supervised learning. So this is, for example, where a baby sees a picture of a cat, and then the mother says, this is a cat. So you are supervising what the image is. You're saying, this image is what we call a cat, and a different image is called a dog. And then the final learning is how we act. It's called reinforcement learning. That is actually the engine behind a lot of the recent AI revolution. It was formulated, including by people like Peter Dayan, for whom we got the Brain Prize at the Gatsby unit. And reinforcement learning simply reinforces behaviors that are more likely to lead to favorable outcomes in the future. If you grab for some people, if you grab for something positive, like you like a strawberry, you're more likely to grab it again. And equally, that can explain many things, for example, why we end up doing lots of doom scrolling, because we constantly get these little dopamine hits, which is what underlies reinforcement learning, okay? Also, it's when reinforcement learning goes wrong, especially with drugs of abuse, that can also lead to addiction. Okay, so it reinforces certain behaviors. If the behavior gives an undesirable outcome, then you are less likely to do the same behavior. Okay, so we're also trying to figure out how the brain does that. Good, okay, so how does the brain do this? Now, it does it by computing in complex neural networks. Now, I'm going to briefly tell you about that, but I just want to make one critical point. The brain is not like this computer. The brain is a computer, but it's not like a conventional PC or Mac that you use. Conventional computers have hardware, and then on top of that, you put software that controls the hardware. In the brain, the software and the hardware are the same thing. They are the neurons that connect to each other in myriad of complex ways. So this is kind of want to give you just a brief snapshot. I know most of you might know this, but there is beautiful complexity here that I think we should appreciate. So the brain is made of neurons. Our human brain has, 80, 90 billion neurons, 150 trillion synaptic connections, and they send little action potentials that travel along incredible speeds, up to 100 meters per second. And so it takes microseconds to communicate from one neuron to the next, and then that's passed on. The messages from one neuron to the next are passed on through synapses that you can see in the bottom corner. That's a chemical transmission, and this is how neurons actually spread information across the brain. Now, this is a reconstruction of a single neuron in the human cortex made with electron microscopy, and the little green points that you see are the synapses, where other neurons make connections onto this particular neuron. This is a pyramidal cell. And each neuron can have up to 10,000 individual connections from other neurons. And in this just tiny chunk of human cortex, 1 millimeter cubed, there are 57,000 neurons and this many, 150 million connections. This is an incredible complexity, and that's just a tiny millimeter cubed of the human brain. Now, to make things more complex, not all cells look alike. There is hundreds, perhaps thousands of different classes of neuron in the brain, in different parts of the brain. And so to understand that complexity is extremely challenging, and all might have slightly different computational roles in the brain. Each neuron talks about 5,000 others. Often when you see pictures of the brain, you see kind of a sparse diagram of connections. This is actually what a tiny little cube of cortex looks like. These neurons are densely interwoven with their dendrites and axons coursing all over each other. It's a real jungle, and to untangle this complexity is one of the real challenges. Okay, and finally, those circuits, because they communicate with action potentials, they give rise to these patterns of activity. It's within these patterns of activity where computation in the brain takes place. So a lot of what we do is trying to understand these dynamics that go on. Now, as you can see from this movie, the blinking is a little indicator that we put, inside the cells, and we can use microscopy to visualize when each neuron is active. This movie is sped up about 10 times. You can see how these patterns are extremely complex, and this is just a tiny field of view that is about 0.5 millimeter across. Okay, so what does understanding the brain actually mean? For me, understanding the brain means connecting across all these different levels of understanding brains, from neurons on one hand to the circuits they form, to the complex neural dynamics of these activity patterns that they generate. And ultimately, those dynamics can be captured in kind of algorithms or computational principles that eventually gives rise to the psychological phenomena that I talked about earlier, inference about the world, deciding what we should do, how we plan, and then how we learn. So it's a hugely complex challenge, and so the only way to do this is to collaborate across the different disciplines, across neuroscience, across data science, because we generate lots of heavy data, machine learning, engineering, and so on, to untangle this. And many of the tools that we use to really probe these circuits and to interrogate those circuits and link them to behavior, we need to use mice. Mice, and mostly mice, occasionally rats, because we have very powerful genetic tools to manipulate and record and label those circuits. The good news is that the mouse brain, the basic anatomy is very similar to the human brain, both at the level of large anatomical structures, but also individual cell classes. You can see two pyramidal cells, those black wiry things there, that are in principle very similar, and there's been numerous studies that have shown that the cell classes, the individual groups of cells across the human and rodent brains, are largely similar. So that's good news. So what we try to do is figure out the mechanisms in rodents and then translate to see if we can find the same principles also in human brains through either non-invasive methods or occasionally by recording in patients with electrodes. So how do we do our science? It's a virtuous cycle between theory and experiment and modeling. So initially we start with a theory or hypothesis on top about how a particular brain region might control or give rise to a particular behavior. Then we generally record activity using different methods. We then construct models of the activity of the neural dynamics, and then we can then manipulate that activity to have causal manipulations. So some of the tools that we use, for example, are the neuropixels probes. These are very dense electrodes from which we can record hundreds, if not thousands, of neurons at the same time, and that really can capture what are these dynamics that are happening when a particular individual is doing a particular behavior. Now, these neuropixel probes have been developed in part at the Sainsbury Wellcome Centre, but funded, crucially, by Wellcome and Gatsby Charity with our partners in the US. And now these neuropixel probes are used globally, and they're also transforming medical applications in humans. So now variants of these neuropixel probes have developed to be able to record from patients, for example, and they are the major component of brain-machine interfaces, where patients, we can use machine learning to train, so the patients can train an artificial network to control, for example, robotic arms or some sort of external object, which is quite transformative for people with paraplegia, for example, who cannot move. Now, another key tool that we use is how we establish causality in the brain. And this is to be able to very precisely manipulate specific groups of neurons in the brain so we can make the link between what's the activity of that group of neurons and the behavior that the individual generates. This approach is called optogenetics. It's a super smart approach where some very smart scientists took little channels from algae, this one here in the picture, modified it slightly. This channel is sensitive to light and it lets ions through. So when you shine light onto neurons that express this protein, neurons will become active. And we have other probes that we can shut neurons down just by shining light. And by putting these channels in specific groups of neurons in the brain, we can specifically manipulate their activity. Here's just one example. So this is activation of neurons in the hypothalamus, which is deep in your brain, that controls aggression. We know animals sometimes have to be aggressive to fend off enemies or to compete. So this is one example. You will see a blue light come on. So this is a mouse in its cage with a rubber glove. Now the light comes on. So we're stimulating aggression circuits. And now the mouse starts attacking a rubber glove. Normally, it completely ignores it. Now the light has been turned off, and the animal behaves as though nothing happened. Now the light will come on again, and you will see what happens. It's looking for an attack point, and it starts attacking the glove again. So this is just an illustration. So this is an illustration of what one can do when one can turn specific circuits on or off, that we can really have a causal relationship between neural activity in a particular brain region and what it does on the animal behavior. Okay, so I'm just going to close by giving 4 examples of how we link circuits to behavior. So the first one is our group leaders, Sonia Hofer and Tiago Branco, have identified circuits that can control fear and bravery in mice. Okay. This is in a structure called the ventral lateral geniculate nucleus. You do not need to know the details. But the behavior that this region controls is a classic fear response or a defensive behavior in rodents. Normally, rodents are afraid and have a defensive behavior to aerial predators. So for example, if a bird of prey is looming and approaches the animal will run away. That's a natural response. And it's been evolutionarily hardwired into those animals because those ones that didn't escape got eaten and hadn't had a chance to reproduce. So we can mimic that behavior in the lab where we loom a dark spot overhead. You will see that in a second. And as soon as that happens, the animal escapes into a little shelter that is infrared. You can't see it here, which it finds safe. So again, the mouse is roaming around the little cage. The loom comes on and the animal escapes. Now, what this structure does, the ventral lateral geniculate nucleus, or VLGN, we can now use these powerful optogenetic tools to activate those neurons in VLGN. And because they suppress other neurons, you can see that when we increase activity in VLGN and you loom the animal never escapes. The fear response is completely abolished. And we can do the opposite experiment, where we give a mild stimulus that normally the animal doesn't find very aversive, it doesn't find it very scary, and then we can increase its fear response, and now it escapes to that stimulus even previously it had not. So we can bidirectionally control the activity in his brain structure, either to increase or decrease fear responses. This structure exists in humans, so this is now a potential entry point in individuals, for example, with anxiety or PTSD, and maybe use deep brain stimulation to see if we can actually interfere and help ameliorate those symptoms. So that's just one example, and I'll give you 3 more very brief examples. Sonja Hofer's lab has found also something very cool. There are circuits deep in the brain in a structure called the median raphe nucleus. It has many different cell types, but one of those cell types does something very amazing. It causes the animals to persevere at whatever it's currently doing, okay? This might be important for things like OCD or compulsion. I'm going to give you an example here of a mouse. The mouse is going to interact with an aversive object. This object, you see, just smelt it, and now the mouse will go into the corner and hide away from this object. The reason why it doesn't like this little black object is because it's coated in a fox urine analog, and mice find this smell intrinsically aversive, and the animals hide in a corner, usually rattle their tail in fear, and they want to avoid it at all costs. Now, let's look what happens when we activate a particular group of neurons in this structure. So the animal sniffs it just like last time, but now, this one here. It sniffs it, we activate the neurons, and instead of the animal running away, the animal comes back and now picks up the object and doesn't want to let go. And as long as the light is on, as long as we are, in this case, silencing these neurons in the median raphe, the animal will keep interacting with this object, even though intrinsically they find it extremely aversive. So we think these circuits control really elemental behavioral strategies, like whether you stick with what you're doing or whether you choose to switch to do something else. I'm pretty sure all of us have found ourselves doom scrolling on our phone occasionally, and at some point you say, okay, that's enough, let's do something else. That switch is controlled by these circuits. And there are other little cell types in this region, then when we activate those, the animals drop whatever they're doing and start doing something else. So it's and those circuits are shared across all mammalian brains, they're not just in mice. So there's a real interesting entry point into understanding, for example, how we can help individuals with obsessive compulsive disorders, where we can maybe undo that compulsion where repetitive behaviors are persist. Okay, a couple more examples. Marcus Stevenson-Jones' lab. Normally, dopamine reinforces rewarding actions. Marcus has found a cool new mechanism that it doesn't just reinforce actions, it just reinforces any behavior that is rare. It's located in this structure called the SNC, the magenta region over there. And we know that that structure is also important and goes wrong in Parkinson's disease. Specifically, those neurons that release dopamine, they die off in Parkinson's, so humans can no longer initiate movements. That's the main symptom. And Marcus's lab found a mechanism by which the brain learns to generate these kind of stereotypical movements, i.e. habit formation, using a different system than the classic rewarding dopamine signal. So this now provides a new therapeutic angle whether we can then rescue those neurons that die off in Parkinson's disease. And finally, work from my lab and Tim Behrens' lab, we have, over the last 10 years or so, come up with theories and found the mechanisms, how neurons connect to each other, such that they can infer the state of the world. If you remember the illusions that I talked to you about earlier, how we fill in, how we imagine a square, and how we learn to imagine the frog, we think that's embedded, that internal model of the world that helps us perceive the world is embedded in the structure of the connections, and specifically in the strength of the connections between the neurons. So now we have theories that we want to test about that, and we know that's important because these internal models of the world are actually what go wrong in disorders, psychiatric disorders such as autism spectrum and psychosis. Okay, last couple of slides about why I think this work is important. Well, I hope what you recognize from the last couple of slides is that just by wanting to understand how neural circuits in different bits of the brain contribute to different parts of behavioral repertoire of animals, we can actually get a little bit closer in finding about circuit entry points for various diseases in the brain too. It is this fundamental understanding of these behavioral processes that we need to build up in order to be able to even cure them in the future. Now, going from left to right, there are certain, how easy is it to target particular circuits and cure certain disorders? There are certain neurodevelopmental disorders which there's a direct link between the gene and the phenotype. For example, certain childhood epilepsies have mutations in certain calcium channels, and we can perhaps do then gene replacement therapy and find drugs to help them. Other disorders are localized to specific regions of the brain. I mentioned to you, for example, Parkinson's disease, where dopamine neurons die off, a specific type of dopamine neuron die off. So we can think of new strategy for doing that. But many of the disorders that are called psychiatric, whether they're mood or affective disorders, so anything from ASD, schizophrenia, anxiety, bipolar disorder, depression, those are difficult to pin down onto individual genes. While there have been clear linkages between genes and those phenotypes, often it's very difficult to link the two. And so those are circuit dysfunctional problems. And so what we need to do is really understand the circuits before we can even begin to start treating them at the circuit level, which is probably where they need to be treated at. And the final point about why our work is important, it comes of a question of origins. So the brain is a hugely complex organ, well, probably the most complex organ in the human known universe for now. And I think understanding the origin of intelligence is one of those big questions. You know, we don't know what the universe is and where is it expanding to. You know, the other big one is what is the origin of life, and I believe that origin of intelligence, of how we can flexibly, how any animal can interact flexibly with the world, is one such problem. And by combining neuroscience and machine learning, these two fields can really feed off each other. We've seen that neuroscience has fed a lot of information into the machine learning and AI fields, and now we hope that AI field will equally feed back a little bit into neuroscience to give us more insight. And it's this interplay of the two that I think will lead to transformative discoveries over the next 10 or 20 years. Okay, I'd like to stop there. Thanks very much.
So you mentioned quite a lot about the increasing use of artificial intelligence and machine learning, and how that's, it's a really exciting area that's been applied to research. Can you tell us a little bit more about how you're applying it and the team are applying it at the Sainsbury Wellcome Centre to neuroscience research?
Yeah, thanks, Georgina. AI. There's probably at least 3 ways by which we use AI to help us understand the brain. So the first one is just tools. We're generating huge data sets, recording from brain activity, mapping circuits, looking at behavior. And AI tools are now helping us rapidly digest those data and interpret those data, including make predictions or hypotheses about what neural activity should look like. So that's the first thing. And we've seen that in many other fields. AI is used to diagnose cancers from MRI images, et cetera, et cetera. So that's a huge accelerant in what we do. Previously, we used to do it manually. Now we can automate many of those things. So that's a major advantage that will just speed things up. That wouldn't necessarily give us insight. It's just going to speed things up. The second way we do it, we train AIs to perform complex behaviors just like humans would or animals would. And then we can dive in and dig in there and say, well, actually, how do they do it? Is it similar to how the brains do it? And can we identify some common principle between artificial agents and real brains? And that is a bidirectional conversation that will kind of inspire. So, for example, how artificial networks, how units in those networks connect to each other might tell us something about how real brain neurons connect to each other. How the information is transformed across the layers might tell us how information is transformed across the different stages in processing in the brain, et cetera, et cetera. And also, what are the learning algorithms that AI agents use, and are those the same learning algorithms that the real brains do? So that's the third one. Is not what we do yet. I know I should just end. This is more of a looking forward question, and that is, imagine we train AIs on like ChatGPT, for example, on all of neuroscience literature or all of scientific literature, And imagine in 5 or 10 years time, there's a super intelligence that is smarter than a typical graduate student or smarter than me. They could actually start generating really meaningful hypotheses and generating new ideas how the brain could work or how a particular circuit could work beyond what we can do right now. Right now, they don't have that capacity. Well, I'll do it two ways. One is finding this link between how artificial agents are structured and how human brains are structured. And that conversation can really accelerate the development of superintelligence, which I think is going to be fun, but based on how you build architectures, how you build learning algorithms to do that. In terms of breakthroughs, I think it's very difficult for me to predict what the future breakthrough will be. I think what I would find very satisfying is if the kind of stuff we do by understanding circuits and how they drive behavior, if they can be translated into something that is useful for humanity. Specifically, if something what we discover in the lab can then be moved on to patients to say, right, we need to stimulate this group of neurons to ameliorate anxiety or PTSD, for example. That would be extremely satisfying. Now, we are not equipped to do the translation. We can seed that. We had a lot of spin-out companies coming from the center. A couple of them are going in that direction. But I think this is something that is worth partnering with. The other question is, as you know, in the UK, to accelerate that, I think the infrastructure in the UK is less good than it is in the US, for example, to go really from discovery reaches into proper translation. And this is something I guess Wellcome could also potentially help with. That's a great question. I mean, if you figure out how dopamine works in reinforcing our, behavior patterns, that is exactly what social media are doing. And they're tapping into actually a particular mechanism. When you do a behavior and you get a little bit more reward than you expected, then you're even more likely to do it again. If you keep getting the same reward, you're going to get bored of it after a while. So that's why you can see in a lot of social media feeds, the content gets a little bit more extreme or a little bit more exciting or whatever it is that releases dopamine that actually keeps you hooked. And that's unfortunately what is actually end up people developing more extreme views, because that's how you push people to click on things, because you always need to go more extreme than the current. So am I responsible for that? I don't know. I blame that on discovery science. I think this is... That's a question of regulation, I think, and whether governments should allow algorithms like that to exist, even though we know they're not beneficial, especially for the younger generations. I don't think that's a question that scientists can actually solve. It's a regulation question. Our job is to discover things, unfortunately. Yeah, thanks for raising that. I think I gave a wrong impression. We don't always start with a theory. Sometimes we start with a lucky finding that we then think about, come up with a theory given the existing literature, and then we test the theory and refine it further, right? So one of the lucky findings is compulsive circuits. That was in a way by chance. We knew this structure was broadly important, but not necessarily for this kind of behavior. And the insight was not to activate this whole region, but rather activate a particular group of cells in this region that is genetically identifiable. And then that revealed this very specific part of the behavior. So this is actually a history of neuroscience is riddled with such findings, where, in the old days, you used to stimulate just one chunk of the brain, like 50 years ago, and there was not much effect. But now, if you stimulate the same chunk of the brain, but a particular group of cells in that chunk of the brain, then you see a really clear behavioral outcome. In motor structures of the brain, it would be the animal could reach or grab or, groom itself, whereas before it just did a jerky reaction that you couldn't really classify as anything meaningful. So it's that resolution that gives us the deeper insight. But hypothesis and experiment are in a virtuous cycle. Where you start on that cycle is unclear, but you need to complete it to generate the deep understanding. Yeah, we wanted to bring more diverse voices into our field. Neuroscience and machine learning, they're tricky fields to get into. So we do this in several different ways. Already, students who are already from underprivileged backgrounds who are already in scientific fields. We bring them in, we partner with social mobility foundations, and then we give them opportunities to really get a head start in that. Equally, we work with schools, because often the problem is that smart kids from underprivileged backgrounds in state schools, they don't want to go into science. They want to do business, medicine, something like that. So it's about working with those kids, getting them excited, and we organize a lot of courses here, or we go to schools, or we go abroad, Kosovo, Romania, Africa, many places in Africa, Ghana, South Africa, etc., where we get kids excited about this stuff. And now, recently, we realized that just us doing this, because we're a relatively small center, is not really penetrating the community so deeply. So now we started teaching teachers how to get kids excited about this, to tell them, so they can tell the kids about careers. So we are training the teachers, and we are now partnering with the Camden STEAM Center that is partly funded by the Google AI Center, and we are giving courses and lectures here in the local community that will also amplify the efforts there.
Thank you so much, Tom, for such an interesting talk and for inspiring us all about the wonderful work of the Sainsbury Wellcome Centre. Hopefully you now have a better understanding of how complex the brain is, the different ways we think about and understand it, how we plan our experiments, the virtuous cycle of research, as well as how the impacts in the future of this type of experiment, and to tell us more about how we can improve human health, and particularly in psychiatric disorders, as we touched upon, and other diseases. So huge round of applause, please, for Tom.
Thank you.